In order to create induced pluripotent stem cells, a large population of reprogrammable somatic cells is required. Many somatic cells have been successfully reprogrammed. However, fibroblast cells are commonly utilized because they can be easily extracted from a patient using a safe and non-invasive skin biopsy and are easy to culture in a lab. Fibroblast cells are somatic cells that produce connective tissue. Morphologically, the cells appear flat, and they secrete collagen and elastic fibers that make up the extracellular matrix. Every cell type in an organism, apart from gametocytes and undifferentiated stem cells, are somatic cells. Each somatic cell has a unique structure and function due to a distinct combination of genes that the cell actively transcribes and translates into functional proteins. The difference in gene expression between different somatic cells is largely due to the structural organization of the chromosomes in each cell. Chromosomes can exist in two forms. The DNA can either be tightly wound around histone proteins to yield heterochromatin, or the DNA can be loosely unwound to form euchromatin. This loosely unwound DNA permits the binding of transcription factors and other transcriptional machinery, such as RNA polymerase, to the gene's promoter region. Once the transcriptional machinery has bound, gene transcription begins, and a molecule known as messenger RNA is made. When the DNA exists in a tightly wound configuration, the DNA is not exposed to the transcription machinery, and transcription does not occur. In this manner, the cell can regulate the genes it does and does not express. In fact, out of the 20 to 25,000 genes of the human genome, a somatic cell will only express a small fraction of the genes at any given time, and the rest of the genes are considered transcriptionally silent. This is the case for the reprogramming factors OCT4, SOX2, KLF4, and CMYC in the fibroblasts. Because these genes are not actively expressed, scientists must induce the expression of these genes artificially using transgenic strategies. There are a number of methods available for artificially overexpressing genes in a cell. The Yamenka group utilized retroviral vectors as the method to deliver the transgenes encoded for the four reprogramming factors OCT4, SOX2, KLF4, and CMYC. The retroviral vectors deliver the four transgenes into the cells. The transgenes are then integrated into the host's genome, thereby permitting its long-term gene expression. Amazingly, when the four transgenes are co-expressed in the same somatic cell, the differentiated cell is reprogrammed into a pluripotent-like state.